Hi, this is Miss Litton, and this is my wonderful 7th period honors review. Say hey. Hey. All right, so we're going to review chapter 24. So the big thing I want, I'm going to put a blank up here, okay? I'm going to put some scientists up here. Could you tell me, or I'll name some scientists, could you tell me what they did? Okay, or what would be harder? Let me do it the harder way. Could you riddle me this? Who is it that found out that the number of A's would be equal to the number of T's and the number of G's would be equal to the number of C's? Who did you say? Chargaff. Okay. All right. So that was one. Who came up with the idea that DNA is a double helix? Who? Watson and Crick. Watson and Crick. Okay, good. Who came up with the idea to shoot X-rays through DNA to look at the pattern that they made?
Hershey and Chase.
No. Rosalind Franklin. And remember, this data was used by Watson and Crick, and so was this data. Remember that? Who worked with viruses to identify that DNA or that nucleic acids are our hereditary material? Yes. Hershey and Chase. Because you would always chase down a Hershey bar. Right? Okay, I'm trying to think if there's any other ones I had you learn. I think those were the only scientists, right? Yep. Okay, so real quickly, I'm just going to show you the picture so I can cement it in your brain. The whole, go. Okay, the whole bacteriophage thing where they label the virus. Remember, bacteriophage means a phage is a virus that infects bacteria. They labeled the DNA with an isotope of phosphorus, and you're going to remember that because base, sugar, phosphate. Okay, and then they labeled the protein with sulfur. Okay, so if you can remember phosphorus, and the other one has to be what they labeled the protein with. That should probably, hopefully, remind you of that. Okay, and they identified that what the transforming agent is that the virus shot into the cell was the nucleic acid, and they know that because they found the radioactive phosphorus inside the bacterium. That's the one where they let it infect it, and then they used a blender to separate the virus from the bacteria, and then they centrifuged it. You have a memory of all that? Okay. Then the next scientists we said Watson and Crick were the ones that came up with the double helix shape. Okay. And they used bond angles, and they also used research from some other scientists. One of those was Chargaff, and he's the one who said A's and T's match and G's and C's match, but he wasn't 100% sure what that meant related to the structure. That's got to stop. And Rosalind Franklin and Maurice Wilkins, they're the ones who shot X-rays through DNA and came up with this pattern, and that said it's a double helix of constant diameter. And we know now that makes sense because adenine has 2 rings in its base, and it always binds with thymine that has 1 ring. So you always have a purine with the pyrimidine, and it keeps your diameter the same. Okay. Okay. Then it's the next thing you really need to be comfortable with is just the structure of DNA. So I think of it as if you unwind it, it looks like a ladder, and you just remember that phosphates and sugars alternate on the sides, and that the nitrogenous bases are in the middle. Okay, so I'm going to just kind of give you an opportunity to test you on this. If I said, what are all the things that sugars are connected to, what could you tell me? What are all the things that bases are connected to, what would you tell me? And what are all the things that phosphates are connected to? Discuss with your bio buddy for a second, and then we'll go over what they are. Okay, what are sugars connected to?
Bases and
Bases and phosphates. So remember how we looked at the sugar like it was a house, like this, right? So it's going to be connected to some base. In this case, I've drawn what? A purine or a pyrimidine? A purine. Okay? And then up here, this is going to be a phosphate, right? And on the other side, it will be connected to another phosphate. You all right with that? Okay, if I ask you what a base is connected to, what would you tell me? A sugar and another base on the other side, right? And so if I drew two hydrogen bonds like this, then you would know what two bases do I have right here? Adenine and thymine. You know that because there's two hydrogen bonds between adenine and thymine. So who is a base connected to? A base is connected to another base and a sugar. And then who are phosphates connected to? Sugar. Only sugars. Does that make sense to you? Okay, now I didn't do this one in class, but let's see if you can handle this. I think you can. You know purines and pyrimidines, right? Right? Okay. So look at this. Donde está esta la diagram? Where are you? I know it's in here. Sorry, delay of game. It's somewhere. Here it is. Okay. Could you label this diagram if I gave it to you? I'm going to tell you like 98% of the world couldn't. Right? So what's the first thing you would tell me to label? Phosphates and sugar. So tell me a shape. Tell me something.
Circle equals phosphate.
Circles equal phosphates. So these right here would be phosphates, yes? What else? And what's the name of the sugar? Deoxyribose. And where does it go? These things? Okay, on both sides. Good. Notice the houses are going two different directions, leading, lagging strand. Remember how we talked about that a little bit? Okay, what else could you tell me to label? Adenine and thymine. Okay, so let me pull out an adenine and a thymine, and you tell me where they go. Okay, adenine and thymine have how many double bonds? Or, sorry. Erase that. How many hydrogen bonds between adenine and thymine? Two. So it would either have to be this up here or this down here, right? Okay, so let's go with this one down here. Which one would be adenine? Which one would be thymine?
Big rectangle.
The big rectangle is adenine. Why? It's a purine, two rings, and thymine would have to be here, right? And we could now label two hydrogen bonds. Okay, what else could we label? Guanine and cytosine. So I'm going to pull these guys out here. And that would have to be wherever you had what? Three hydrogen bonds. So here are 3 hydrogen bonds. Which is which?
Guanine is bigger.
Guanine would be the bigger ones. Oh, wait. And cytosine would be here. Yes? Okay, do you feel smart? Okay, so there's not a single label on that, but you were able to identify all the structures. You should feel very proud. That's AP right there, what you just did. I didn't do it in class, but this really shows that you understood because you had to deduce, right? You had to use your critical thinking in order to figure everything out. All right, now is DNA built the same as RNA? No, because DNA is what? Double-stranded, and RNA is single-stranded. Okay, and DNA has the sugar deoxyribose, whereas RNA has the sugar ribose. And you don't have any thymine, instead you would have uracil. Okay, but before we move on to that, let's talk about DNA replication. Okay, DNA replication. So when would I want to do DNA replication? Yeah, if I'm doing mitosis and meiosis. And it's got a particular name when you talk about DNA replication. It's called replication. What is that? Semi-conservative replication. Because each old strand serves as a template for a new strand. Okay, so let's form our DNA. Ready? Here we go. The DNA has to unwind and unzip. Helicase. Remembering that because we look like an H. Okay. One strand would be the leading strand, and that has to do with the enzyme we're going to use. What's the name? Say my name. DNA polymerase, right? So it can only read in one direction. And the analogy I gave in one of the classes, I don't know, maybe I gave them in all three, but if you're at a street, and if I went out here on Canaan and I looked down Canaan, and if I was an enzyme and I was bringing in complementary bases, I have to face trap it. So as I look down Canaan, I'm in the center divider. if I look to the left, I'd have oncoming traffic, right? So I could go right, boom, 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 and just attach on the free nucleotides going right along. But this way, on the right-hand side, traffic's going with me, right? You've learned that in your driving. Okay, traffic's going with me. So I would have to back up, go down Keenan a little bit, and then turn around so I would have oncoming traffic. And then I'd have to go back down a little bit farther towards the replication fork and turn around so I would have oncoming traffic. That's the lagging strand. That's why when we're here and we use DNA polymerase, we go boom, boom, boom. That's on our leading strand. And then here we start at our elbow and we go boom, boom, boom. Okay? So let's go again. DNA unwinds, unzips, helicase, DNA polymerase, boom, 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 boom. Now there's more to the story, but you do have to stitch the phosphate sugar backbone together, so we say what? Ligase. Good. Then these would twist up, and I would have more hands, right? I would be this because I replicate it, right? You with me on that? Then these would be sister chromatids held together by a centromere that separates during anaphase. Perfect. Now, that is if I'm copying my whole cookbook, because I have my house in Kauai, right? I wish. So I need a cookbook in that house too, so I need all of it. But if all I'm doing is baking a pie, I don't want to copy the whole cookbook. I want to copy one section of it, and that process is not DNA replication. This is called transcription. And I need to get an RNA copy of my DNA. And for that process, forming this RNA, okay, so I'm at this point, I'm using this DNA to build this RNA. But since DNA is double-stranded and RNA is single-stranded, I only need one of the strands. So once again, I'm going to unwind and unzip, helicase, but I'm going to take this slow strand where we went boom, boom, boom. I'm going to put him away. I don't need him anymore. And now instead of using DNA polymerase, I'm going to use RNA polymerase. And I'm going to boom, boom, boom again. This time where there's an adenine, I'm going to bring in a uracil on my nucleotide. If there's a thymine, I'm going to bring in a adenine. If there's a guanine, I'm going to bring in a cytosine. If there's a cytosine, I'm going to bring in a guanine. If there's A, I bring in U. If there's T, I bring in A. Yes, so you still have adenine. So I make my mRNA, make your mRNA, peel it off, set it aside, go get your DNA, zip it up, put it away, go get your mRNA. Now, this mRNA, I can process that, I can manipulate it, I can edit it so I get different proteins. When I do that, I cut out what? Introns or exons? Introns. I cut out the introns before it leaves the nucleus. I also protect either end with a cap and a tail. The cap is 7-methylguanosine, the tail is 100 to 200 adenines. And that's what you see going on. Here you're making RNA from the DNA. You'll learn about the promoter later. And here you can see they've put on a cap and a tail, and they're cutting out the introns before it leaves the nucleus. So from one strand of DNA, I make one strand of mRNA, but I can manipulate the mRNA so the end product comes out different. And we talked about that in our analogy. I could replace apples with peaches, right? I can make a crustless pie if I wanted to. It was all one original pie recipe in my nucleus, but I can modify the mRNA and end up getting a different product. And that's what you're doing right there. Okay, and then the next part is the translation part. And the translation part is actually using your recipe to make your pie. And your workbench for all of that is what? Ribosome. Now, in the ribosome, you will have rRNA. So that's one of the 3 RNAs that you have. This right here is called what? tRNA. And what is it transferring? Amino acid. And then we have mRNA. This is our 3rd kind of RNA that we have. The only RNA that gets translated into a protein is the mRNA, the messenger RNA, just this. The other ones are workers. The other one's like, okay, fine, build it on me. Okay, here, this is what you need to build a protein. The only one that gets translated is the mRNA. Every 3 bases on the mRNA, is called a what? Codon. And it is complementary to 3 bases on the tRNA called, I don't know what I'm writing here, on the tRNA called a what? Okay. or this, what I labeled, okay? Now, are these two complementary to each other? If you can't see this, this says GCG. What would be the anticodon that it would be complementary to? CGC. What amino acid would that tRNA carry? What do we need to look up? The codon or the anticodon? Codon. So the codon is what? GCG. So let's go to our codon chart. Here's the first one is G, the next one is C, and then the third one is G. GCG is alanine. So that was the amino acid it carried. So I can use the codon chart to tell me what the tRNA is bringing me. I'm using the codon chart, the codon is on the mRNA, to tell me what the tRNA is bringing me. Now, the next thing about that is when you look on a codon, okay, that this is showing you 2 binding sites, but technically there are 3. Okay, this side right here is called the A site, and this is the P site, and then a third site on here would be E for exit. But basically what happens is that you have this new guy land right here on the A site. Now it's got other names, but I remember it because it has a single amino acid on it. That's how I think of it, as the A site. And it's sitting right next to another tRNA who has the whole chain on it. And as this ribosome shifts, it drops this chain off on the new guy, on proline, and then when the ribosome shifts, then this tRNA gets put not in the P site, but in the E site, and then he will exit. And then this ribosome will have moved down one, a new tRNA will land, and he gets dumped on. What basically happens is the new guy lands and gets dumped on. The new guy lands and gets dumped on. And the guy that just dumped on the new guy, he gets put in the E site, and then he leaves. And that's going to go, 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 go until you reach a stop codon. So if we looked at it here, there is a small and large ribosomal subunit. So the small unit right here, here is the first tRNA to land. That is tRNA 1. It is always going to carry with it methionine because the first codon is always AUG. Now that methionine might get snipped out later, but for initial, that's always the first one. So he lands, then the large ribosomal subunit. Now look right here. What are our 3 sites we have right here? A, P, and E. So the next guy is going to land here, and then as this ribosome moves this way, he will dump methionine off onto the new guy, and because it shifts, then he will be put into the E site for exit. And this will keep going and keep going. So here we can see this guy's going to land here, then he's going to shift. Oh, as you can see in this diagram, he's going to shift and dump it on the new guy, and this guy's going to move into the E site. So that's happening again and again until you reach a what? Stop codon. And the stop codon, then it will shift onto nothing. It can't bind anything. It just shifts and dumps it off. And then you will get your chain of amino acids, which is a protein, and the small and large ribosomal subunit will fall apart. This mRNA might get used by another ribosome. And literally those ribosomes can get on there and like read it, and then another one can get on and read it, and another one can get on there and read it. So it can be used over and over again. And we'll talk about the control of that in the next chapter. So putting that all together, what you want to be able to do is if I give you a strand of DNA, you have to tell me, you need to be able to tell me what the complementary DNA is and the mRNA you would get from it. If I give you the mRNA, guess what I want you to be able to tell me? The DNA that it came from or the tRNA it could bind with. You see how that works? Okay, and you don't change anything into anything, right? What you do is you say who is complementary. They always have the same dance partners, okay? So for DNA, who is complementary? If I was going to make more DNA and I was going to do replication, and I would have obviously 2 strands of DNA, boom, 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 2 strands, booming, okay? Who would be complementary here to this DNA? Yes, C, exactly. All right? But if I was going to use that same strand to make mRNA, what would it be? Okay, I want it to be a different color. Okay, G, I'm listening. U. Because when you make RNA, tRNA is still T, right? Would be who we would think would be complementary. There just aren't any T's in RNA, so I have to use U's. So every 3 bases on there is called a what? Codon. So I have a codon here, and here, and here. Do you agree with that? Okay, so then there would be complementary anticodons. What would be the complementary anticodon to this one? A. Okay. So these would be on tRNAs, and each of these would carry an amino acid. I could find out what that amino acid is by looking up what? The codon chart. So what would I look up for this first one right here? GUA, is that accurate? Where am I?
CAU. No. Do you look at the initial code?
What do you look up?
So the tRNA.
The anticodon will tell you what's on the tRNA, but you don't actually need it to find out what the amino acid is. I'm trying to find where we are writing. Where are we writing? Oh, I found it. Okay, so here you go. Hey. Okay, so what do I need to look up for the first one? GUA. So I'm looking up GUA. It doesn't matter. GU, anything is always going to be what? Valine. Good. VAL. Okay, what's the next thing I look up? AUC. AUC is isoleucine. What's the next thing I look up? Okay, CUC doesn't matter, it's leucine. Right? Okay, so that's how you can determine the sequence of amino acids in a protein. Okay, yes?
I don't know if this matters, but is the anticodon like a part of the tRNA, or is it like attached to it like a
Let me show you that. So the tRNA looks like this. Okay, all of this is RNA. It loops around, it has particular loops on it, it actually kind of folds back on itself. Kind of like plastic wrap does when you stick it to itself. It'll fold back on itself, and this loop right here is the anticodon, those 3.
So it is.
Okay? Yes?
So for the other one, the page, the yellow page you just did.
Yes, this page right here.
So how come you look up the codon for the mRNA, not the tRNA?
I'm looking up the codon to find the amino acid. Okay? Because this is the chart. The mRNA is what determines what tRNA will land and then what amino acid. That is actually the code.
Mm-hmm.
Where did it get the code from?
From
the DNA. Mm-hmm.
Okay.
So this is the code that you have from the DNA. The tRNA will land wherever it's complementary.
Oh, okay. So then you take the mRNA because the tRNA's
Well, I know what
opposite.
tRNA is going to land on that.
'Cause
Not
it's
opposite, complementary.
complementary, so
Yeah.
that's why they bond together.
Yes.
Okay.
Yeah. G with C, right?
Okay, that makes sense.
Yeah. Yes.
Is there any time where we are going to be using our anticodons? Like to code for
Well, you'll use your anticodons, but you really work off the codon because that's the part that gets manipulated. That's the part that's getting read. So that's what they give you the chart for. Yes.
For your first tRNA, will it ever carry something that is not CAU? Or like, are those the anticodons that it will always carry?
No, I just threw these letters up. I didn't even actually know what they coded for.
But like, for
What?
that specific one, will the bases ever change on it?
Oh, there's actually something called the wobble hypothesis where you might end up something. There's nothing I'm going to ask you about. Yeah. Yeah. And you look up the codons. What's interesting is how it doesn't matter if you have a point mutation here in the 3rd codon, right? It doesn't matter. You're still going to get what? leucine. But if you have a point mutation in the first of it, you'll get something else, right? So here is CUU is leucine, but if you accidentally get a point mutation, you get AUU, then you get isoleucine. So usually the third one is the least concerning. You can tell that wherever there's blocks. You know, the key one is the first two of the codon. Okay? Now, let's talk about those mistakes, because that was the next part of it, is we talked about, okay, that your genetic material has to store information, but it has to be stable so you can pass it on, but it has to mutate, and mutate. Okay. And so we want to know what a mutation is, a change in the sequence of bases within a gene. Change the bases, you have a mutation. You know that because if you change the bases, you're going to change the mRNA. If you change the mRNA, you're going to change the tRNA that lands on it. And if you change the tRNA that's going to land on it, you're going to change the amino acid, right? Okay, unless it's those ones where we looked at it. If it's the third one, sometimes it doesn't matter. You end up getting the same thing. And what kind of mutation is that called? Silent mutation, okay? So we also talked about things that cause mutations. These are all things that could cause mutations. So you want to avoid anything that's a mutagen, right? Or exposure to something that could wreck your DNA. We also talked about, ironically, you can change the DNA. It can change itself by these kind of genes. What kind of genes are these called? Transposons. And they're called what genes? Jumping genes. And literally, they excise themselves from one part of the genome and put themselves somewhere else. So that can change the DNA. And then we talked about frame shift mutations, right? And I used simple 3-letter words. And when you do a frame shift mutation, it impacts every codon from there on out. So it's not like if I had just replaced the E with an A, okay? Could we still read this? The big fat cat ate the big rat, right? We could still read it and make that sense with that. But I didn't replace it. I eliminated the E. So now my first codon, instead of being THE, my first codon is THB, which then throws off the next one and the next one, frame shift. So I've wrecked all of them. So frame shift mutations are really bad because they wreck every single mutation. Now, a point mutation that we looked at here is when you just change one letter. So I changed the third letter from a G to an A. Didn't matter because I still got tyrosine, so I'm good. My amino acid didn't change. And that's somewhat typical in your third letter of your codon. Not always, but sometimes. Here we changed the third letter and it did make a difference. It coded for a stop codon right in the middle of our chain of amino acids, so that's not good. Here we changed the first letter, usually that's not good. It coded for a totally different amino acid. Are you okay, sweetheart? Okay, coded for a totally amino acid, a different one, and that would be super bad. Hashtag super bad. Okay, and then this is a page where I kind of summarized all those mutations, and you probably put it in your notes, I'm sure. Just kind of summarize them. Okay, and then that was the end. Okay, so I think I have a practice quiz I can give you if you'd like to take one. Would that be fun? But I'm going to go ahead and stop this now. Have a great evening. Hopefully it's not too late. And if you're hungry, what would we want to tell them? Yes, have a piece of toast. Okay, tell them goodbye. That was not very enthusiastic, but we do love you. We do love you most of the time.